Okay, welcome back everyone. So we now have the final talk of today's session, and our speaker is Professor Giuseppe Vitiello from Salerno University, who will tell us about quantum origin of collective behaviors in biological systems.
Sì, mi dai 10 minuti prima delle 35. Okay, good afternoon to everybody. We had a busy day, and I would like to thank the organizers, and especially Luca Bonifante, for inviting me to talk about let's say, quantum field theory applied to living matter. But before that, let me, this is a sad thing which I would like to remember Francesco Guerra, who has been one of the leader Italian physicists. He has been working in Princeton on Euclidean field theory in 1978. And he has been a prorector at the University of Salerno, involved in many administrative, but especially scientific activity. He's lost this is something which I've been the first student that took the degree with him. We were both very young, and now is the last Saturday we had. I wanted to remember him because he was really outstanding. He taught me to use mathematics trying to understand physics and not for itself, which sometimes it happens because mathematics is beautiful, but sometimes we remain trapped in the mathematical formats. So for me it has been a sad. Okay, so let me start with, this is, the title is Quantum Origin of Collective Behaviors in Biological Systems. If there is any quantum origin. This is a story which starts a long time ago, very long time ago, and especially because it was the same Schrödinger who was giving a series of talks which were collected successively in a book with his life, which maybe you know it has been a very popular book. And he was asking how it is possible that solely based on biochemistry was a rule, is a random rule, so everything is based on concentration of reactive substance. How is possible to justify, let me use this word, those who are regularities in biological systems? So regularities only in the average emerging from the statistical mechanism is not enough to explain the enigmatic biological stability. These words are from the book I told you, What is Life, on page 78 and 47. And he was stressing that the attempt to explain the biological functional stability in terms of the regularities of statistical origin would be the classical physicist. We are at the beginning of 1940, so the classical physicist expectation put in those years these words, that far from being trivial is wrong. Of course, he was Erwin Schrödinger, he couldn't say something in so strong words. And so this in some sense has been the trigger for many people to try to understand if there is any possibility to treat living matter, which after all is matter, like is matter, is stable or ferromagnet or a proton, if there is any way to use the formalism that was just born in quantum mechanics in some fruitful way. And actually, history has not been like that in biology, because this is taken from Springer Nature of 2020. Is a paper by Bizzarri of approaching complex diseases, where Giuliani, Alessandro Giuliani, told about ISS in Rome, Institute of Studies Applied to Biophysics. They were saying that the paradigm which is commonly used in biology is that a gene A provokes phenomenon, disease, phenotypic trait, etc., B, by means of a pathway C. And I observed that the existence of a single explanatory layer is in sharp contrast with what we know about the complex structural systems, where multi-layer causality is at work. But the situation is, to them, is still something like that. And then there are other references which I skipped because of time. But for example, there was a time in which everything was concentrated on studying the genome, and the promise of a druggable genome set forth by the completion of human genome sequencing with consequent opening of a practically infinite horizon of development on new drugs failed. Something very fundamental went wrong. So the situation is not really appealing from the simply minded biology approach, biophysics, biochemistry approach. Then on this part, there was Herbert Friedrich, who in 1968 wrote a paper, one paper, then there were other ones. The title was Long-Range Coherence of Energy Storage in Biological Systems. This was in the International Journal of Quantum Chemistry. This paper was really a revolution. Please think that in those same years, Ilya Prigogine came out with his dissipative systems analysis, and these joint states, let's say, were quite fruitful. In fact, several people started to consider the possibility to use quantum theories in living matter. Among these possibilities, there were let me first say this. Oh, sorry. Okay, doesn't matter. How will go back? Yeah. So among these attempts, there was a paper by Mironio Mezzawa and Luigi Maria Ricciardi in 1967 in Naples, where Mezzawa was spending a visiting professorship, in which they were trying to understand brain and proposing the brain as a many-body system, namely applying to brain studies, so to neuroscience, quantum field theory as you do normally in many-body physics. Consider also that those years were very nice years for the physics, in particular for quantum field theory. Superconductivity, the BCS theory was just in those years. So something was going on. Now, why quantum field theory and not just quantum mechanics? There is a difference which we know quite well, everybody of us knows. In quantum mechanics, the Stone von Neumann theorem tells you that all the representations of the canonical or anti-canonical commutation relations are unitarily equivalent, which means physically equivalent. So there is no point in choosing one or the other one. It's just a matter of what you prefer or what is better for your computation. But the physics content is the same. Now, this theorem fails in quantum field theory. And this is characteristic feature of quantum field theory, a really basic feature. Namely, in quantum field theory, since the quantum hypothesis of a phenomenon theorem in quantum mechanics fails, namely you have infinitely many degrees of freedom, then you have infinitely many unitarily non-equivalent representations of the canonical commutation relations, commutation or anti-commutation relations. This point is crucial. It's crucial to many problems of physics. Just think of many body physics, where you have phase transitions, a system in two different phases, in two different dynamical regimes, is described as in two different, namely unitarily non-equivalent representations. And you have the problem of phase transition, of course, which you cannot deal with just in quantum mechanics. But for living matter, this is a crucial point, because biologists tell us that the biological system is alive when it is at non-equilibrium. What that means? It means that the biological system goes through infinitely many dynamical regimes. If it does not do like that, it is a dead system, it's not a living system. So a living system can only be described by a manifold of representations which are physically different, each one from the other one. And this is given by, this possibility is realized in quantum field theory. So this, I insist on this point because it's a very important point. What means that? It means that we can have in quantum field theory, not in quantum mechanics, the mechanism of spontaneous breakdown of symmetry. Everybody knows nowadays is it's a basic, how to say, tool to deal with several, many problems. Now, spontaneous breakdown of symmetry, also this morning, the Goldstone modes that were mentioned. We have the Goldstone theorem, which says that as an effect of spontaneous breakdown of symmetry, there is a dynamical realization, a dynamical appearance of a long-range correlations in the system. So you can explain, for example, a ferromagnet through the existence of a long-range correlations, which are the Goldstone modes, the number of Goldstone modes. In that case, you call them magnons. In the case of a crystal, you call them the phonons. So, spontaneous breakdown of symmetry means that in the system, made of many components, you may have phase correlations. Order arises in a dynamical way. Please consider that order, contrarily to everyday language, is a lack of symmetry. Order is when we can make a distinction between things. Symmetry, it means that everything is equal to itself. The system, the invariance of a theory, means that the equations are symmetric under transformation of a certain group of symmetry indeed. So this is the starting point of Mezzao and Ricciardi proposal to study, for example, the brain, as I told you, as a many-body system. Okay, I told you already about these things. There is one important point which came up also in the discussion of this morning. How it happens that we have a quantum background, but we have a classical behavior. You see, a diamond is forever. There is the advertisement, the commercial. A diamond is a collection of atoms and phonons in a given order, because we have a breakdown of translational symmetry in that case. And such an object, a diamond, you buy as a macroscopic classical object. So how it is possible to reconcile the quantum description of a crystal that we do have, and is very good. I mean, there is a lot of results, and it's not a matter of discussion today how we describe a crystal by using quantum field theory. How is possible that such a quantum description gives us a classical behavior? This happens because the Goldstone mode the long-range correlations, if you think of them in terms of quanta, associated quanta through the Broglie relation, they condense in the ground state. The ground state is a coherent state. A coherent state generates an order parameter. This order parameter, like magnetization, like density, this order parameter is a classical field, although is born from a quantum dynamics. The magnetization of the needle in the compass is not fluctuating. Is a classical, determines the classical behavior of the needle in the compass. But the origin is a quantum origin. This is a crucial step, the transition from the quantum level to the classical behavior. This transition is not the naive classical limit which we have in quantum mechanics, where you send h bar to 0 and then thank you very much, you obtain a classical behavior. But in quantum field theory, you obtain a classical behavior out of a quantum dynamics just because of coherence, just because h bar is different from 0. This is a beautiful transition mechanism which allows the transition from the quantum dynamics at microscopic level to the higher macroscopic behavior. And this is what happens in the living matter. This is, in some sense, the realization of what Schrödinger was asking for at this time. So, the stability of the order parameter, I already told you about this, and then we might even talk of a macroscopic quantum state in such a meaning, according to what I just told you about the order parameter. After that, it happened to me, you must be lucky in life, there is nothing to say about, because you must meet the right people at the right time, which happened to me. It happened to me to meet indeed Herbert Frobik and Humezawa too. And in Milano in 1983, with Emilio Del Giudice, Silvia Dogli and Marcello Milani, we started to apply the machinery of quantum field theory by using path integral, because it's very difficult to write a Lagrangian for a piece of living matter. With the path integral, you might just ask to the Lagrangian some kind of symmetries and then work out all what the machinery of functional integral techniques. So we studied the spontaneous breakdown of symmetry indeed and boson condensation as I told you, in biology in a series of papers. Here are the titles of a couple of them. And please notice, this paper, we had to publish this paper not on Journal of Biology, because biologists had not read mathematics, had not read quantum field theory. And so we had to find where to publish such things. And I remember I was calling Giorgio Verizzi, asking him, Giorgio, help us, where we have to send these papers. And of course, the papers available at that moment for physicists studying, I've been studying neutrino mixing. By the way, there will be on Friday talk on neutrino mixing by using quantum field theory. So, where nuclear physics, B, and there was the section field and statistics, and there was physics letters A, of course, and these papers were published in those journals for physicists, of course, but that is the story. Or for example, on physical review letters, like this one on water as a free electrical dipole laser with Emilio Del Giudice, Giuliano Percalata, and myself. So this was the start of my involvement in this kind of subject. And we obtained something which is, to me, is still today, now, at this moment, to me, I do not ask us to be the same to you, is striking. If you talk with a biologist, he will tell you that inside the cell there is the cytoskeleton. The cytoskeleton is made by microtubules. These microtubules are crucial because all the metabolism happens along microtubules. They form they elongate, they become shorter. There is a mechanism they call treadmilling, namely they lose something and they reconstruct and so on. So these they are very well observed, and they found that their inner diameter is about 14.5 for 15 with experimental errors, of course, nanometers. Now, simply basing on the, in some sense, quite abstract. The only data were how much it is the dipole of a water molecule, you obtain exactly that for the microtubules, the inner diameter is 146 angstrom, namely 14.6 nanometers, which is exactly the number which people observe in biology. Now, how we did such a thing? This is interesting for a physicist, so I can tell you in a different, with a different audience, I will skip this point. You see, here we apply the Anderson-Lipes-Kegel mechanism. Namely, since water molecule carries a dipole, an electrical dipole moment, it gets polarized. Fortunately, water forgets. Namely, polarization has a very short lifetime. And so you have many dynamical successive polarization and depolarization and so on. But while you have, in such a short time in which you have polarized a domain of water, of course, then if there is an electromagnetic excitation, well, we know that it should propagate through filaments according to its mechanism, getting a mass. And we applied exactly, the paper is there, you can look at, is a 1986 paper, nuclear physics, so physicists can read the nuclear physics journal. You find that just computing those things and putting those numbers according to the matter you are dealing with, you find such a number. There is no computation at this moment, as far as I know, about the computation from a dynamical framework of the inner diameter of such an important object like a microtubulus. The justification that the biologists give for such a size is that the tubulin, which is the macromolecule which runs around the microtubule and actually forms the microtubule, the bonds, the chemical bonds, are such that it can turn in such a way to produce 14.6 nanometer. But this, according to a physical point of view, is not the cause, but is the effect of what happens. The dynamics creates the microtubule according to such a general principle of Anderson-Lipes-Kegel mechanism. The dynamics creates such a microtubulus. I mean, as you see, inside the field E is a non-zero, outside is equal to 0, so you have a gradients, and you have gradient forces which attract or repel molecules, and you construct such a coating which at the end of the story is microtubulus. So you see how powerful can be a physical theory with respect to the study of living matter. So this is one of the results, and we have nowadays is a common subject studying water as a two-component system, namely ordered domains, flickering ordered domains, and non-ordered ones. So there are papers as ones I show you these titles here. And then there is something one should say, because you see, we are always very good, but sometimes we make a confusion, a mess. The word coherence, if you are in a framework of quantum mechanics, you talk of decoherence, but the decoherence has nothing to do with what in quantum field theory you mean by coherent state. A diamond melts at 3,000, where is at 3,550 centigrades at atmospheric pressure. So coherent states in quantum field theory, coherent states because the diamond is a coherent state of phons keeping atoms in their positions. So coherent states are completely stable. I mean, a lifetime much longer than our lifetime, in some sense, if you allow me to say this. And there are many. For example, the magnetization in cobalt is lost at 1075 centigrades. This is coherent state, and so on. Okay, this is just to. Now, it happens that this coating, this behavior of water allows to water, okay, 10 minutes, thank you, allows water to influence, to promote, if you want, biochemical reaction. So one effect you may have in such a situation is that is a water-mediated correlation in DNA enzyme interactions. Something like this. You see, this is the usual charge interacting through exchange of a photon, a gamma, and in a similar way you have a dipolar wave, water, which mediates the interaction between the DNA and enzyme. So this kind of situation is you find a paper by DNA chiral spinal hydration by Luke McDermott, Panzerius and other people, published in 17, and they show, is an experimental paper, they show the discovery of a chiral water superstructure surrounding the DNA under ambient condition, I mean a condition of a living system. And this has been the first observation of a chiral spine of hydration temperated by biomolecules at room temperature. In all this description of what they did this year. I did not spend time because time is going on to finish. And they observe in their paper that a change in the hydration state of water can lead to dramatic change to DNA structure. I do not know. But my question, I'm tempted, let's say it this way, I'm tempted to put the question, DNA is like that because of biochemical bonds, or is it like that because those bonds are the effect, of course biochemically altered, are the effect of the water hydration? I do not know, I cannot say that something like that, but there is such a kind of situation which you find here. This is a picture which appeared in those papers I'm telling you. Then we think, well, but biochemical molecules is quite heavy molecules. Indeed, you can consider gramicidin. This is a paper on somewhere, I do not remember, there is after Nature something. Gramicidin, let me see, is composed by 15 amino acids. Well, this group here, are the authors, they have been showing something like the double slit experiment with such a heavy, big stuff producing, I mean, it's a delicate experiment, but they have shown, the weight is 1,882 atomic mass units. So it's a really heavy object. So these objects, they do behave like quantum objects. This morning we were talking of quantum and classical again. We might not expect such kind of things, but they do happen as we observe. These are not hypotheses. This is all of the description of the experiment and the fitting with classical and quantum prevision, and the quantum was. And then you have something, for example, in the a weak olfactory stimulus This is in a small animal like a rat. Mice. Activates about 10 to 3 neurons in the olfactory bulb. This stimulates the excitation of 10 to 6, 1 million, of neurons and the inhibitory activity of 10 to 7, 10 millions of neurons, which propagates in between 5 and 10 milliseconds over a distance of about 10 millimeters, even if the average length of the axon are of about 1 millimeter. You see what means long-range excitation? Long with respect to the typical object you have to deal with, in this case, the cell and the axon length. And the synaptic propagation time is about 10 times longer. There is something more about neurons and axons and glial cells and all that. In the brain, most part of the brain is 90%, more than 90% water. So maybe water is not just a solvent, as a biochemist or a biologist would say. Water is an actor. Of course, in a highly nonlinear process between water and the constituents, biochemical constituents. So, at this point, if I have still some minutes, let me review the dilemma put by Carl Lashley at the beginning of 40s. Lashley has been one of the masters in modern neuroscience. He says, here is the dilemma. Nerve impulses are transmitted from cell to cell through definite intercellular connections. Yet all behavior seems to be determined by mass of excitations within general fields. Look at the words he uses. It is very of activity. Without regard to particular nerve cells. What sort of nervous organization might be capable of responding to a pattern of excitation without a limited specialized path of conduction? Still today, people, neuroscientists, they talk of wiring in the brain. There is wiring, axons, dendrites, and all that. But those are not the only, constituents responsible of a long-range correlation among neurons in the times you do observe. You see, this is a crucial point. For example, of course, Carl Pribram has been one of the pioneers in introducing in neuroscience, as you know, after the laser construction, he proposed that in the brain something like an holographic dynamics could go on, just to justify how it's possible in those short time. You see, if you compute, Walter Freeman did such a computation, a chemical, a neurotransmitter, as you call them, a neurotransmitter, so a chemical molecule, is much slow with respect to the time you do observe in the brain when some part of the brain enters in resonance with another part of the brain far away. Let me jump on this. This is a very common picture. You see all these blobs of neurons. These are coherent domains of neurons. The coherence, the idea, the proposal here, is that coherence is promoted, is facilitated by the coherent structure of the water component, which of course is flickering, which is crucial because we have to change the possibility to change our mind according to the outside input, otherwise we would be crystals. These are the two guys, Umezawa and Freeman, which I've been so lucky to meet and to work with them on these problems, apart from the ones with Umezawa about physics. And then you might construct what is called the dissipative quantum model of the brain. Umezawa in 1967, I told you, he was proposing this brain model without dissipation, however, at the time. And he was writing, in any material in condensed matter physics, any particular information is carried by certain ordered pattern, maintained by certain long-range correlation, mediated by massless quanta. And he was talking of Wilson model, of course. it looked to me that this is the only way to memorize some information. Memory is a printed pattern of order supported by long-range correlations. This morning we had a talk, a beautiful talk about memory through condensation of coherent Gaussian modes. Okay, this kind of subject gave me the possibility, you see, for example, he writes, first of all, at which level should the brain be studied and described? In other words, is it essential to know the behavior in time of any single neuron in order to understand the behavior of natural brains? Probably the answer is negative. The behavior of any single neuron should not be significant for the functioning of the whole brain. Otherwise, higher and higher degree of malfunctioning should be observed. You see, this is the physics approach, the physicist approach. Okay, I jump because time is over. Spontaneous breakdown of symmetry, I told you already so much. And Walter Freeman, he was a neuroscientist. In his lab, he was observing chaotic dynamics in the neuron population. This chaotic dynamics is the chaotic dynamics of trajectories through many representations, coherent states of different density, through which the brain goes in its functional activity. You can show that just by using your mathematics we share, and there are the publications there where you can show this. And this is a picture to this trajectory in the space of the representations, which are, according to the Keller theorem, chaotic deterministic trajectory, classical trajectories. I think I could stop here, but there is this final, maybe thinking, which is not the think, the thought, is not the one we use when we show that Pythagoras theorem is true, going from an hypothesis with one well-defined necessary series of steps, uniquely defined. That is trivial, a machine can do that. The thought, which means invention, which means creation, which means finding a new solution to an old problem. Maybe that thinking means to go to wonder. In Italian we have a nice word, errare, which means going wandering, but it means also making mistakes. So thinking is making mistakes, is wandering. This is a lesson which in days, I should say dark days, as one we are living just in this moment, we should think about and not rely just on machine which we give them to solve our problems. They work according to a thinking which is not wandering, is not making mistakes. Making mistakes is a privilege of humans, so we should keep that. And this was, okay, of course, one of our fathers from Neumann. The mathematical or logical language truly used by the central neuron system is characterized by less logical and arithmetical depth than what we are normally used to. This is 1958. We require exquisite numerical precision over many logical steps to achieve what the brains accomplish in very few short steps. Okay, let me, this is closeness. A living system is an open system. If you close the system, you kill the system. This Caravaggio and this Narcissus sit the closed circle of the arms and then reflects into water. And of course, this letter, Narcissus dies. And about 15 or 16 years after, you see, this is open system. You see, the law of geometric optics tell you that if you see in a mirror the face of someone, she, in this case is a she, is looking at you. So this is a metaphor to me of an open system. She is watching at her observers and vice versa. This mirror is situated in, I mean, if she, we should not see her face if she was looking at herself. This is the point. So this is an open system in this life, of course. I will not tell you anything about the fractal structure in living matter, which we found in our model, and which these are experimental findings by Freeman. This log plot and so on, but forget about it. Okay, thank you very much. Sorry for
Okay, questions?
Thanks for the very interesting talk. And just one comment that biological systems are very complex than universe. Universe has more symmetries than biological systems. I'm curious if there is any progress on applying quantum field theory to understand cancer biology, because this is where people are, majority of biological people are interested to understand how cancer spreads from.
Okay, thank you. Your question is very welcome, because just in these days, after a meeting in Sweden, there I met a colleague of us, an experimentalist who in New York is at the City College, University, something like that. In this group are studying samples of living tissue in cancer situation and in normal situation. And they find a relation between the water, because there is a lot of water in any case, between the water, they studied through X radiation, reflection, and interference. So the water, which is there, seems to behave in different ways to give a different spectral picture in the case of tumor tissue and healthy tissue. So we decided to remain in contact to see if such a difference is such that the tumor, I mean by tuning the water state, the tumor is enhanced or balanced or hopefully decreased as a bad situation by tuning the water state and by measuring. This would mean a dynamical approach, not a biochemical approach, where you kill everything in, as you do in normal chemotherapy, and at the end of the story you killed the person. But this would be very much interesting. Thank you for such, you gave me the pleasure to tell you this.
Steph?
Thank you for the very stimulating talk. I want you just to make a comment, because your talk reminded me of a study that actually I managed to find. I don't know if you know that it's been 5 years ago about the study between the connection between the filaments in the universe, which are very similar to the neurostructure in the brain. There is a collaboration between an astrophysicist from INAF in Bologna and a neuroscientist in Verona. I was wondering if there is some connection between this study and what you have been talking about.
You see, one of the pleasures of a theoretical physicist working with quantum field theory is that he studies structures. Then you put a different amount of ingredients and you get your own pie. So there might be such a connection, as you say. After all, this string-like object in of cosmological interest, let's say, I remember Tom Peeble when he was studying such kind of things, and we were talking also with him about such possible connections. I cannot say anything more than this. There might be, but there are so basic dynamical structure that my expectation is that there is a connection. But this is, if it is like that, it would be, let me say, aesthetically beautiful, because we have really basic pictures able to describe, according to the local specific ingredients, completely different situations, in completely different systems, but after all, we have some kind of a universal understanding. Yes?
Maybe one last question.
Thank you for the talk. This is a much more mundane question about the chirality in the spine of the DNA. I just wondered, are you saying that this electromagnetic imprint in the spine is an imprint of the nucleotide sequence in a way that is going to control where the polymerase comes and attaches itself to the double helix? Is that the suggestion?
Suggestion is already too strong word. Is a suspect. But the paper I was telling you about, what happens? Oh, but
It's left.
the paper I was is about this group, which is a well-known and well-organized group in. Sorry, this one. Yeah, this one they studied, but they were saying that was the first time they saw a change in the hydration state. That the hydration state can lead a change dramatic, they write, they word, in the DNA structure. Now, what that means? To me, the experiment means that the DNA, which is always embedded in water, every student in biology, they know that they cannot take out all the water, something always remains, the hydration water indeed. The hydration water, but they add the DNA in the water, and this is what they see and what they say. But such a dramatic change if you touch water, to me, means a nonlinear dynamical system. It means that they live not separately. Water is not the bath, is not a solvent, is an actor. So, who starts first, you are asking, in some sense. And this was also my question. I do not know. But since the system is just one, water and, which is not the point of view of biologists, of a biochemist, they consider biochemist and water. And they make a soup. I think, as a physicist, that you cannot do that. You have a bottle element, that is just one single system. And you have to study the nonlinear coupling between the two. Who is the first? I mean, maybe we do not even have to ask such a question, but they interact each other. This is why, in the case of tumor tissue, I do hope that by tuning the state of water in some way, I do not know, you might introduce an element which non-linearly, which means we don't know how, might convince the cell which is going away, the tumor cell, to go back, making again an healthy tissue. I do not know, but this is the situation which. Sorry if I cannot give you answer on this point, but I would be very glad if I could.
Okay, so let's thank Giuseppe Vitiello. Okay, so the scientific session of the day is finished. Scientific part